Hello, Geert. First of all, I want to thank you for taking the time today to, really get into the details of your theories and issues with this vaccination program, with this pandemic, and the SARS coronavirus. So thank you for taking the time.
You're very welcome. Thanks for having me.
And for our audience that's watching, and as you know, you are in Brussels, Belgium. I am in Austin, Texas. But through the magic of filmmaking, we're sort of sitting in the same room virtually because I wanted to put out a video that really feels more intimate because we're going to get very intimate on this conversation. So to begin with, I have done several shows now on the different interviews you have done over the last almost half a year, last several months. You put out a video very early on from your own social media page warning the WHO that there was a real problem with the approach towards the SARS coronavirus or SARS-CoV-2.
Dear colleagues at the WHO, my name is Geert Vanden Bosche. My background is veterinary medicine. I'm a certified expert in microbiology and infectious diseases. I have a PhD in virology, and I have a long-standing career in human vaccinology. I'm urging you to immediately open the scientific debate on how human interventions in the COVID-19 pandemic are currently driving viral immune escape. I'm urging you to invite me for a scientific hearing open to the public and to scientists all over the world on this very topic. Ignoring or denying the impact of stringent infection prevention measures combined with mass vaccination using prophylactic vaccines is a colossal blunder. Please do listen to my cry of distress, and let's first and foremost deliberate on a scientifically justified strategy to mitigate the tsunami of morbidity and lethality that is now threatening us. And let's meanwhile devise a strategy to eradicate the steadily emerging highly infectious variants. On behalf of humanity, I sincerely thank you for considering my call.
To begin with, this time, I want to start out with sort of your background. What is it specifically about your experiences that is giving you a different perspective than we're hearing from other world-renowned scientists?
Well, Del, I think it has, in fact, more to do with what I decided back 10 or 12 years ago, where I decided I would take a completely different approach to science. But it started all with my intention that I would start all of a sudden to look at the science not as, an objective in its own right, but as a tool to solve problems. Because I was really very much bothered, having worked in both academia and in industry, I started thinking more and more about these two different worlds that both start with P. The world of the publications, that's what counts in academia, and the world of products, that's what counts in industry. And in between, everybody talks about translational medicine, but it's very, very rare, in fact, that we find solutions, that we use the science to really solve problems. And of course, my background is veterinary medicine. That's very broad, that's very diversified. And I had a chance during my career to touch upon several different fields. I specialized in virology, in vaccinology. I learned my vaccinology, in fact, in industry. That's also the place where I learned immunology. I was teaching zoonosis, so infectious diseases that can be transferred from animals to humans at the university. I have a long-standing interest in evolutionary biology, and all this has to do with interactions between the pathogen and the host. So the host immune system, and understanding these interactions is very, very important. And then what's also very important is to understand the evolutionary capacity that a pathogen has when it is put under widespread immune pressure. That is something you don't have in clinical studies, for example, right? This is the population effect. And so this simply illustrates that you have to be able to draw from all these several different fields. I was so fed up that we put many products in the pipeline in industry without even understanding how they interact, how they work exactly. And I was so fed up in academia that all what counts is publications. We need to connect both, because otherwise we are in a situation right now where we put out products without understanding what's going on, without understanding the pandemic, and you know my point. I'm 200% convinced that this is going to lead to a catastrophe, to a disaster.
Geert, you worked for Gavi on the Ebola vaccine program. You've worked with the Bill and Melinda Gates Foundation. Now, obviously, you didn't start out by deciding to put your career in jeopardy as a world-renowned, vaccine maker, as a scientist, as a professor. You must have reached out to your peers at the WHO, who I know you've worked with before, at Bill and Melinda Gates Foundation, at Gavi. How did you reach out, and what was the response?
Well, Del, I've reached out at least 3 times to, I would say, all the global health authorities, a number of public health authorities, CDC, NIH, the Bill and Melinda Gates Foundation, WHO, of course. And I got maybe 1 or 2 anonymous responses. Maybe I said this in an earlier interview, and I'm not going to disclose his name, but it is certainly one, if not the most famous vaccinologist on this globe, who told me, Geert, you are right, these vaccines are basically just going to breed variants, but nobody is going to listen to you because you go against the mainstream, right? I mean, imagine. It is just, I have no words for this. Were you surprised when you got no response? Is this the first time you've reached out with an issue? No, I was not surprised, no. The Ebola story, I mean, it was the same, but it was a small scale. It was just the countries in West Africa.
What was your job on the Ebola vaccine?
Well, I was coordinating the Ebola program, and part of this was the vaccines. It was not just the vaccines, was also about a number of measures that needed to be taken, to control more or less the Ebola crisis in collaboration, of course, and with WHO and UNICEF and CDC and strengthening health measures, etc. So it was a collaboration between all these international health authorities. But of course, since I came from the vaccine field, I was very much interested in the vaccines that would be deployed and that would be used.
And what was that issue when it came to evaluating the effectiveness of the vaccine and the safety of the vaccine?
Basically, it came down to the naive interpretation of those who conducted the study that the incubation time of Ebola was, well, I don't remember exactly 10 or 12 days, and that therefore they would not need to start the clinical observations before so many days after the vaccination, after having identified the index case, and then you have the contacts that were all vaccinated. The endpoint was vaccine efficacy after, so many days after the vaccination. But the vaccine that was used was a live vector that everybody knows, and I come from the adjuvant field as well, is very, very strong in inducing inflammatory cytokines. if you study the pathogenesis of Ebola, you will find out that people die of a cytokine storm, of a huge inflammatory cytokine storm. So what do you think happens if people who are incubating this because you identified the index case and then you were immediately vaccinating the contacts who potentially were already in the incubation phase and would have been the ideal controls. So these people are already incubating a disease that leads to inflammatory cytokine storm. And then you come with a vaccine that is well known to stimulate pro-inflammatory cytokines. So these people, those who were infected and were vaccinated, they simply didn't make it till day 10 before they started the observation.
Wait a minute. So let me get this straight. They basically said we know the incubation to be of Ebola to be about 10 days. Therefore, when we find the index case, the person we know that's infected, they went and vaccinated everybody that had come in contact with them.
Yeah, ring vaccination.
Right, ring vaccination, ring around the index case. But they said, let's not collect any data for the first 10 days because we know that if anything's going to happen inside that 10 days, it's, it shouldn't count. And what you're saying is you gave an inflammatory vaccine